Good afternoon, and thank you for joining today's IBEAM webinar. I'm Morgan from IBEAM Technology, and I'm very excited to have you here. This webinar will highlight how advanced in vivo imaging technologies are transforming cardiovascular disease research, with a particular focus on real-time visualization of the beating heart in living systems. Today, you will discover how intravital microscopy enables researchers to directly observe cellular and vascular dynamics, including lipid accumulation, thrombus formation, and immune cell interactions, providing deeper insights into disease progression at the single-cell level. Next, we are pleased to welcome Olivia Kim, Technical Application Specialist at IBEAM Technology. In this session titled Seeing the Beating Heart in Vivo for Cardiovascular Disease Research, she will demonstrate how intravital imaging can be applied to three representative cardiovascular disease models: acute myocardial infarction, thrombosis, and high-fat diet models, and how real-time in vivo imaging can support more precise and efficient research in cardiovascular disease. Please don't hesitate to submit your question in the Q&A box at any time during the presentation. We will do our best to answer as many as possible during the Q&A session at the end. And if you'd like to learn more about today's speakers or explore additional applications through our homepage, please check the Resource tab at the bottom of your screen at any time during the webinar. So, Olivia, let's begin.
Good afternoon. I'm Olivia, a technical application scientist at IBEAM Technology. Today, I'm pleased to share our work on real-time cellular level in vivo imaging using intravital microscopy, and to introduce how this technology enables dynamic visualization of cellular level changes in cardiac disease models. Today's presentation is organized as follows. First, I will provide a brief overview of intravital microscopy and IBEAM Technology's imaging platform. Next, I will present several research applications in the heart using our intravital microscopy system. And finally, I will conclude the presentation. To begin, I will briefly introduce the fundamentals of intravital microscopy and highlight several key features of IBEAM's imaging platform. So intravital microscopy is a high-resolution imaging technique that allows for real-time observation of cellular and tissue-level dynamics within living organisms. Traditional imaging methods such as in vitro or ex vivo typically rely on fixed tissue slides or cultured cells. As a result, they lack the ability to capture real-time biological and physiological changes and do not fully reflect the complexity of the in vivo environment. In vivo imaging modalities such as PET or MRI are valuable for analyzing physiological changes at the tissue or organ level, but they lack the resolution needed to visualize cellular level events. In contrast, intravital microscopy fills this gap. It allows researchers to continuously monitor drug distribution, cellular interactions, immune responses, and disease progression directly within a living subject. This unique combination of cellular resolution and real-time tracking provides a more accurate and physiologically relevant understanding of biological processes. As I mentioned earlier, conventional techniques only yield rough, low-resolution data since they capture images at the whole body or whole organ level. To overcome these limitations, we have developed a more advanced intravital microscope that enables researchers to track cellular dynamics, monitor drug distribution, and assess therapeutic effects even in challenging organs such as the lung and heart. A defining strength of iBeam's intravital imaging system lies in its clear departure from conventional in vivo approaches. Traditional methods typically require sacrificing separate animals at each time point, which inevitably introduces substantial inter-animal variability. In contrast, our system allows longitudinal imaging within a single living subject at defined intervals, dramatically improving consistency and reliability. Beyond static image acquisition, it enables real-time, dynamic visualization of biological processes as they unfold, offering a far richer and more accurate perspective on complex physiology. To truly understand how our body functions, we need to observe what is happening inside, down to the level of individual cells. This is especially important when studying the immune system, cancer, or how drugs are distributed and act within the body. Intravital microscopy makes this possible by allowing us to visualize these dynamic processes in real time within a living animal. Our system, in particular, enables us to track how drugs travel through the body and how they interact with the cells. In addition, by leveraging transgenic mouse models expressing fluorescent proteins along with fluorescently labeled antibodies, research can selectively visualize specific cell populations or molecular targets in vivo. With multi-channel imaging capabilities, different labels can be detected simultaneously, enabling the concurrent observation of multiple cell types or biological processes within the same tissue environment. This multiplexed approach greatly enhances our ability to study complex cellular interactions in real time. For example, on the left, you can see data from sepsis model, where inflammation leads to vascular blockage in the lungs. On the right, we demonstrate how our imaging system can track exosomes, anti-cancer drug distribution, and transplanted cells as they move through the body. These types of cell-level observations are enabled together with fluorescent labeling strategies that allow specific targets to be visualized in vivo. iBeam Technologies' intravital microscope is designed as all-in-one system, making them easier for users to operate. These systems are simple to install and maintain, allowing researchers to focus entirely on imaging and experiments without limitations from space or environment. Our platform provides an ultra-fast polygon scanner, real-time temperature monitoring system, motion compensation, and AI-based denoising software, enabling high-resolution imaging of diverse organs in living animals. First, because the organs are observed in a living state, their motion caused by respiration and cardiac activity has made it extremely difficult to obtain high-quality and stable images using conventional microscopy. However, with iBeam's unique motion compensation algorithm, we are able to correct these physiological movements and acquire high-resolution, artifact-free images in real time. Second, the real-time temperature monitoring system ensures the body temperature is continuously tracked and adjusted if needed throughout long imaging sessions. This not only minimizes physiological variability, but also maintains an ethical experimental environment. Lastly, when imaging at ultra-high speeds, the data can contain substantial noise, making quantitative analysis challenging. To address this, our AI denoiser software effectively removes high-frequency noise, allowing researchers to obtain clean, high-fidelity images suitable for accurate quantitative analysis. Now let me go into more detail about the components of our platform I mentioned earlier. Real-time observation of cellular dynamics is essential for understanding biological processes in vivo. In the living body, red blood cells and immune cells move extremely rapidly, making it difficult for conventional microscopes to capture these fast cellular events as they occur. To overcome this limitation, iBeam Technology has implemented an ultra-high-speed scanning system utilizing both a polygon scanner and a galvanometric mirror. By integrating these two scanning mechanisms, we are able to precisely visualize the rapid motion of cells in real time. The video shown here is an example of real-time imaging of fast-flowing red blood cells in the beating heart. Using our system, we can capture 50 frames per second at 512 by 512 resolution, enabling accurate visualization and analysis of high-velocity cellular dynamics. The second component is our motion compensation technology. Because all imaging is performed in a living mouse, physiological movements can cause substantial motion artifacts, especially in the heart and lung imaging, where there is constant motion due to heartbeat and respiration. If you look at the image on the slide, the captured image appears blurred and difficult to interpret due to continuous cardiac motion. However, after applying motion compensation, these movements are corrected and the blood vessels and cellular signals become much clearer, enabling more accurate observation. Thus, by applying a motion compensation algorithm implanted in our imaging engine, we can effectively correct these movements and obtain high-resolution, motion-stabilized images. The final component I would like to introduce is our AI denoising technology. When performing ultra-high-speed imaging to capture rapid dynamics, some level of signal noise is unavoidable. In particular, when imaging continuously moving signals such as blood flow, the raw data often contains substantial noise, making it difficult to use for quantitative analysis. To address this, we apply an AI-based denoising algorithm that effectively removes noise while preserving underlying biological structures. As you can see here, comparing the raw video on the left with the denoise results on the right, the vascular structures and blood flow signals become much clearer and more defined. This goes beyond simply improving visual quality. It enables more accurate identification of vessel boundaries and help distinguish true biological signals from noise, ultimately leading to more reliable analysis. As a result, we can precisely track and quantify rapid cellular dynamics that were previously difficult to analyze using conventional methods. This AI-driven approach has also been highlighted in Nature Methods, supporting its scientific validity and applicability for advanced in vivo imaging. Therefore, for highly dynamic tissues such as the heart, a combination of high-speed imaging, motion compensation system, and AI-based denoising is essential to obtain truly interpretable and reliable data. IBM Technologies' intravital microscopes are designed not only for short-term studies, but also for long-term imaging applications. To improve stability and reduce variability, we provide a range of windows and chambers that allow both short-term and long-term imaging in the same animal, resulting in more reliable data. On this slide, you can see a variety of our windows and chambers for different anatomical sites. We have developed customized devices for abdominal organs, as well as for the heart, lung, and brain, each optimized according to the physiological characteristics and surgical requirements of the target organ. For abdominal organs, longitudinal imaging is possible for approximately 2 to 4 weeks. For the heart and the lung, because the procedure requires opening the thoracic cavity, in vivo imaging is typically visible for 2 to 4 hours, while the animal remains physiologically stable. In contrast, for brain imaging, surgical intervention is minimal aside from the initial implementation of the cranial window. And once the mouse has recovered, long-term imaging for over a year is possible without significant additional surgical burden. If there is a particular research interest in a specific organ, we can also develop and optimize customized windows and chambers tailored to the study needs. So using the technologies I have described, our platform enables real-time visualization across multiple organs and a wide range of biological processes. This includes various immune responses, cancer metastasis, the efficacy of anti-cancer therapeutics, and even the mechanisms underlying disease progression. Moreover, our system allows researchers to monitor the in vivo biodistribution and therapeutic effects of nanoparticles, peptides, and other drug candidates with high spatial temporal resolution. For example, we can observe T cells migrating from the vasculature into the tumor tissues, as well as accumulation of collagen-rich fibers in the liver through second harmonic generation without the need for any additional staining or labeling. In addition, we can visualize how anti-cancer nanoparticles traffic toward tumor sites over time, or how the behavior of immune cells responding to inflammation changes following the administration of immune modulatory agents. Importantly, all of these processes can be monitored longitudinally in a single mouse, allowing for long-term within subject observation of dynamic biological events. So from here, I like to walk you through several intravital microscopy applications in the heart, starting from immune cells and blood flow, followed by acute myocardial infarction, then Rose-Bengal-induced photothrombosis, and finally epicardial fat accumulation. The first application I would like to show is real-time cardiac imaging, which allows us to directly visualize immune cell dynamics and blood flow in the beating heart. On the left, you can see a mouse mounted for heart imaging under the microscope. As shown here, the animal's respiration is maintained using a ventilator, and a tissue motion stabilizer is connected to the vacuum-based imaging chamber to gently stabilize the surface of the beating heart. In addition, a rectal probe continuously monitors body temperature and provides real-time feedback to maintain physiological stability throughout imaging sessions. With setup, we are able to perform stable intravital imaging of the heart in a living animal. Now moving on to the imaging results. In this experiment, we used lice MGFB mouse to visualize immune cell dynamics in the heart. Blood vessels were labeled by fluorescently conjugated CD31 antibody, and blood flow was tracked using labeled red blood cells. As you can see, both red blood cells and immune cells are moving through the vessels at very high speeds. This highlights one of the key advantages of our intravital imaging system, where it allows us to directly visualize rapidly moving cells in real time. For the heart imaging, we use the device called vacuum-based imaging chamber. The heart is positioned and gently held at the center of this chamber. This chamber is connected to the tissue motion stabilizer, TMS, which applies negative pressure to stabilize the heart surface in a smooth and controlled manner. Typically, the pressure is maintained in the range of 820 to 850 millibar. In addition, a ventilator is used to maintain stable respiration throughout the imaging process. Now I will show you a video of the heart imaging surgery procedure. So first, the mouse is intubated with a catheter. Then the mouse is mounted on the stage in the imaging room. This is the tissue motion stabilizer, and the ventilator is connected for stable respiration. The skin is cut, and the ribs are removed to expose the heart. Chamber is positioned to stabilize the heart, and after that, the door is closed, and the stage is adjusted for optimal positioning. Finally, imaging is performed. Next, moving on to our next application, acute myocardial infarction, or AMI, remains one of the leading causes of death worldwide. To better understand this, we established an intravital imaging model of myocardial infarction. This model is based on a ligation-based ischemia reperfusion approach, in which a coronary vessel is temporarily occluded and then reopened to restore blood flow. In this model, after inducing myocardial infarction, we observe changes in blood flow, alterations in immune cell distribution, and changes in lymphatic vessels within the heart. To investigate this, we designed the experiments as follows. First, we used type 2 GFP transgenic mice to visualize blood vessels, allowing us to monitor both blood flow and immune cell dynamics. Blood flow was tracked using labeled red blood cells, and neutrophils were visualized using Ly6G antibodies. We divided the animals into two groups, a sham group and an acute myocardial infarction group. This allowed us to assess changes in blood flow and immune cell infiltration following infarction. First, this shows a comparison of blood flow imaging between the sham group and infarction model. The green represents vasculature and in the transgenic mouse. The red indicates red blood cells, and the blue corresponds to neutrophils. In the sham condition on the right, red blood cells are flowing normally within the vessels, and neutrophils in the blue are barely observed. However, the infarction model on the right, blood flow appears to be completely halted, and there is clear increase in neutrophil signals within the vessels. In addition, some neutrophils can be seen extravasating out of the vessels. Based on these observations, we can conclude that the vascular barrier function is altered following infarction. When we examine this in a large mosaic image, we can clearly see that neutrophil signals are much more widely and strongly distributed under infarction conditions. To quantitatively analyze the increase in neutrophils, we measured fluorescence intensity across groups. Compared to the sham group, the infarction model shows approximately a 6-fold increase in neutrophil signal. So then we raise a question: is this increase in neutrophils simply due to changes in blood vessels and blood flow, or could lymphatic dysfunctions also play a role? To follow up on this, we use PROX1 GFP transgenic mice to visualize lymphatic vessels. In this image, the green signal represents lymphatic vessels and the red signal represents blood vessels, and the blue signal corresponds to neutrophils. Consistent with our previous findings, we observed a clear increase in the infarction model compared to the sham group. Lymphatic vessels in the infarction model appears thinner, particularly in regions with high neutrophil accumulation. Based on these observations, we suggest that the increase in immune cells in the heart following infarction is not solely due to changes in the blood vessels or blood flow, but may also be associated with alterations in lymphatic vessels, potentially leading to impaired immune cell clearance. The next model we used is a photothrombosis model. In this model, rose bangle is injected and activated by targeted light illumination, generating reactive oxygen species that induce endothelial damage and platelet aggregation, leading to localized thrombosis formation. Using this model, we were able to observe changes in blood flow velocity and platelet accumulation in real time. Specifically, we aim to test whether rose bangle can induce thrombosis in the heart and consequently alter blood flow. We also examined whether platelets accumulate within the blood vessels. First, we imaged the heart before and after laser irradiation following injection of rose bangle using a 561 nanometer laser. In this video, the blue represents blood vessels. The red signal represents rose bangle circulating in the bloodstream, and the green signal represents platelets. In the first video, taken before laser irradiation, platelets are seen flowing rapidly through the vessels. However, after laser irradiation, as shown in the second and the third videos, platelet flow becomes progressively disrupted and platelets become to accumulate within the vessels. These observations indicate that thrombosis is induced by Rose-Bengal in combination with laser irradiation. To confirm that this effect is specifically due to Rose-Bengal and not simply caused by laser irradiation alone, we performed control experiments using a conventional fluorescent dye with a similar wavelength. So to validate our findings, we performed a control experiment using FSD555, a fluorescent dye with a similar excitation wavelength to Rose-Bengal. The tissue was irradiated under the same laser conditions, and platelets were stained to assess whether they accumulate within the blood vessels. When comparing the image before laser irradiation and after repeated laser exposure, we observed no noticeable changes in platelet flow and even in microvasculature. There were no signs of platelet aggregation or flow disturbance. These results confirm that thrombosis was specifically induced by Rose-Bengal rather than by the laser irradiation alone. Obesity and metabolic disorders have become a major global health concerns, and lipid accumulation around the heart is known to be closely associated with cardiovascular disease. For the next application, we investigated lipid accumulation in the heart using a high-fat diet-induced hyperlipidemia mouse model. Using this model, we examined how lipids accumulate in the cardiac tissue and how this affects the surrounding vasculature and microenvironment through intravital imaging. Here we used B6 mice and fed them a high-fat diet for 8 weeks. To assess lipid droplet accumulation, we stained the lipids using SF44 dye and labeled the blood vessels with CD31 antibody, followed by intravital imaging. In this image, the red signal indicates blood vessels, and the purple signal represents lipid droplets. When comparing the control group with the 8-week high-fat diet group, lipid droplets are barely observed in the controlled conditions. However, after 8 weeks of high-fat diet, a substantial accumulation of lipid droplets is clearly visible. Based on these findings, we further investigated whether a high-fat diet induces not only lipid accumulation in the heart, but also additional changes. Therefore, we also examined the associated immune responses. In this experiment, we used lysed MGFP transgenic mice and fed them a high-fat diet for 8 weeks. Afterwards, we analyzed immune cells, shown in green, and lipid droplets shown in white within the heart. As you can see in the video on the left, a large number of lipid droplets are accumulated on the surface of the heart. Moving deeper into the tissues, immune cells can be observed accumulating in the deeper regions. The image on the right shows a projection from the surface to deeper layers, allowing us to visualize both the surface lipid droplets and the deeper immune cell distribution at a glance. As you can see, prolonged exposure to a high-fat diet leads to a gradual increase in lipid droplet accumulation. And by later time points, we observed a higher number of infiltrating immune cells compared to the 4-week stage. This suggests that progressive lipid deposition can trigger a range of cellular responses within the cardiac microenvironment. In this way, iBeam's intravital microscopy platform enables far more than simple cell observation. It allows researchers to visualize a wide range of biological responses that accompany disease progression or therapeutic intervention, providing a deeper and more dynamic understanding of in vivo physiology. To conclude today's presentation, I would like to briefly summarize the key points we discussed. iBeam's intravital microscopy platform offered far more than single time point imaging. Through a variety of specialized windows, chambers, and organ-specific approaches, including those designed for highly dynamic organs, we enable researchers to access regions that were previously difficult or even impossible to visualize in vivo. At the same time, integrate a system that maintain body temperature and support optimal physiological conditions ensure that the data obtained are truly representative of in vivo biology, allowing for high quality, reliable imaging across a wide range of applications. Also, from basic scale system designed for small animals like mice and hamsters to larger series that can be even applied to non-human primates, our portfolio covers a wide spectrum without size limitations. Through this diverse set of intravital imaging platforms, we are not merely observing cells. We are gaining the ability to visualize the dynamic, living behaviors of cells within their native environment. When establishing animal disease models, there are often many variables, and severity can vary significantly between individual animals. As a result, a large number of animals are typically required to obtain reliable data. However, as we've seen today, intravital microscopy allows us to analyze the same animal before and after treatment. This approach minimizes inter-animal variability and enables us to track changes within the same subject, ultimately improving the reliability of the data. Today, I have shown how intravital microscopy can be applied to various cardiac disease models, including acute myocardial infarction, thrombosis, and high-fat diet-induced models. Importantly, intravital microscopy is not limited to a single organ or disease. It is a highly versatile platform that enables direct observation of cellular dynamics across a wide range of tissues and organs in vivo. Moreover, as shown in this slide, numerous studies utilizing the I-beam Technologies intravital microscopy platform have been published in international peer-reviewed journals, and this list continues to expand. Finally, we have collaborated with many universities worldwide and research institutions, as well as pharmaceutical and biotech companies. This concludes my presentation. Thank you for your attention, and please feel free to ask any questions.
Thank you, Olivia, for the excellent presentation. So we will now open our Q&A session and wrap up today's webinar. So I think there are two questions already. So question number one is, for long-term imaging, do you keep the mouse on the microscope continuously?
It depends on the experimental design. In many cases, imaging is performed at specific time points, such as 2, 6, 12, and 24 hours. If the intervals between time points are relatively short, you can maintain the mouse on the stage by providing additional anesthesia as needed. However, for longer intervals, typically beyond 12 hours, the mouse is returned to its cage for recovery, then re-anesthetized for imaging at the next time point. So both injectable anesthesia and inhalation anesthesia can be used, depending on the experimental requirements.
Okay, thank you. And the next question is about, how do you label blood vessels or other cells for imaging?
Currently, we use antibodies conjugated with fluorescent dyes for labeling, and these are administered via intravenous injection. If there is a specific target of interest, users can conjugate the appropriate antibody with a fluorescent dye themselves. Or if the process is challenging, we also provide in vivo labeling kits that can simplify this step.
Thank you, Olivia. Is there any questions? We are waiting a moment. Okay, so thank you everyone for your questions and active participation. So before we conclude, we'd like to share an upcoming opportunity. On May On May 20, IBIM will host a global cardiovascular webinar featuring Dr. Mikkel, whose research was recognized among the top 10 cardio-oncology papers worldwide. The session will highlight studies conducted using our IBIM series. Be sure to stay tuned for this exciting event. The webinar recording will soon be available on our YouTube channel. If you have any additional questions or if we weren't able to address your inquiry during the Q&A session, please feel free to reach out to us at information@ibimtech.com. Thank you again for joining us today, and we look forward to seeing you at our next webinar. Thank you.